Hello, everybody. Starting with our physical control agents, and I mentioned that there was one mechanical control, which we can easily just put under physical control, so you don't have to, think of that being separate. Also, there is another table in your lecture handout for this week that will allow you to consolidate the next several slides into one table. So you can easily kind of do the compare and contrast of the different techniques, and then how it works or how effective it is, and then the actual method. So let's start with heat. So you probably already know that like we can boil water, right? You might have a boil water order. So if there's been some compromising event that is resulting in potentially human fecal bacteria migrating into the water supply, then you might have a boil order. So essentially, we know that we can boil water and eliminate or kill most of the pathogens that would be in that water. So you probably already know heat is a good thing. Or an autoclave. I mentioned autoclave last time. So that machine that we have in a microbiology lab that goes up to 120 degrees Celsius. So, all right, let's take a look at this. Heat. Elevated temperatures, so when it gets warmer, we're thinking about it being microbicidal. So essentially, we are killing microorganisms. Is it sterile? Not necessarily. It can still have the endospores. So doing a pretty good job at eliminating the vegetative cells of our microbes, but not necessarily those endospores. Lower temperature. So I know heat, we're always thinking about it getting warmer, but if we're low on that range, so we're thinking about refrigerator temperature, then we have essentially a situation where we are slowing the growth of microorganisms. So it's microbistatic. So I recently had the situation where some grated cheese went bad quicker than I thought it would. And usually when our cheese goes bad, it's because there's penicillium or actually some kind of fungal pathogen that's occurring. So if you pull out a brick of cheese and you see like a green fuzzy colony on it, so that's a fungus. So essentially I pulled out this grated cheese. Now we have an older dog and she doesn't like to eat her food. And now she needs some water on her food because she's having a tough time eating her kibble, her dry food. So we put water on it, and then I've been in this bag of cheese with probably my dirty little hand, putting cheese on her food twice a day. So after about 6 or 7 days, I noticed that this cheese started to smell kind of fungusy. So while that cheese is in the refrigerator and it's reducing the amount of microorganisms that's going to grow, me putting my dirty little hand in it all the time was probably not a good idea. So just wanted to impress upon you that our food is likely not sterile or not for very long, right? So if you get something out of a can, it has been canned, high temperature and pressure. So yes, the food is sterile, but as soon as you pull it out, there's spores in the air of fungi that can be landing on your food, and there we go, right? So, okay. So refrigerator temperatures, microbistatic. All right, moist heat. So hot water, boiling water, or steam. And we're thinking about this being between 60 and 135 degrees Celsius is a really great way to kill most of our vegetative cells of our microorganisms, but not endospores. Endospores are going to require a much hotter temperature. So we're thinking about dry heat like incinerating. We're thinking about, this is hot air, open flame. So 160 degrees Celsius to several thousand degrees Celsius. Okay, so mode of action and relative effectiveness of moist heat. So moist heat, okay, so we can operate at lower temperature and short exposure times to achieve the same effectiveness as dry heat. So the moisture is going to aid. And what we know about autoclaves is that temperature can go up because of the use of steam to increase the pressure. And the reason why moist heat works is because essentially it is going to cause clotting and denaturation of proteins, so destroying proteins. Okay, dry heat. So we can dehydrate the cell. So in lab, we've got this special little tool called an inoculating loop, and we're going to flame it or incinerate it until it's completely orange, and let it cool off, and we know that we've removed anything on it. So for those of you in the on-campus lab, once you're taking that cooled off inoculating loop and sinking it into a culture of bacteria and inoculating whatever it is that you want to inoculate, whether that's an agar slant or a petri dish or nutrient broth, well, now you have this big old gob of bacteria on that loop. And then what do we need to do? We need to flame it. And so one of the things that's going to happen is we're going to dehydrate that cell, and it's going to cease existing because we've destroyed its metabolic functioning. But we also can denature or destroy the proteins that are in that or on those bacteria on your inoculating loop. So there's a couple of other things. At very high temperatures, we can see we can increase the stability of some protein conformations. So essentially we need the higher temperatures to destroy those proteins. And then at very high temperature, we can oxidize cells and essentially turn them to ashes. Sounds kind of evil. All right, so here is our perfectly folded quaternary structure of a protein. And so when we heat it up, we are going to first change the active sites so that we are no longer able to do the enzymatic functioning if it's an enzyme. And eventually then what we're going to do is it's going to unfold. And so that's what we call denaturation is essentially we are destroying the properties that it has, the active sites. So then it can no longer accept the substrate, and the enzyme is inactive. And so then it is going to essentially kind of unfold and become just like a linear type of protein, which is changing its function. Okay, so here we see comparison of times and temperatures to achieve sterilization with moist and dry heat. So that moist heat, take a look at temperatures of 121, 125, 134. It doesn't take that long to sterilize, but dry heat is going to take a much longer amount of time simply because we are going through the process of desiccating the cells and affecting the metabolic functioning, and then finally that protein denaturation. Okay, so let's go back to this idea of what are we killing when we're doing this particular type of control method. So in particularly our moist type of control, or those very lengthy dry control methods, we are going to kill our endospores, and we know that they exhibit the greatest resistance. So remember, that's our standard. We want to get rid of the endospores. If we're getting rid of the endospores, then we're getting rid of all the vegetative microorganisms as well. So the destruction of the spores requires temperatures above boiling. So it kind of gives you an idea of the effectiveness of just normal everyday boiling on your stove. Sure, if you have a boil order, you are going to kill coliform bacteria. It's not likely that you're going to have endospores in your drinking water. So that's kind of what's going on with that. You can see the vegetative cells, generally speaking, are going to be more sensitive, but even, a vegetative cell of different kinds of, we'll talk about bacteria, can have different levels of resistance. So that kind of makes sense. Just really depends on your goal. So it comes back to that. All right. So a little more information about boiling. Boiling water, yes, can disinfect. Okay, so it's going to quickly decontaminate items in the clinic and at home. So you might have been in the generation where your parents had to boil your baby bottles, and the nipples on those baby bottles to ensure that they were free of microorganisms. So we know that single processing at 100 degrees Celsius is not going to kill all the resistant cells. So it's only going to be relied on for disinfection, right? So we're going to reduce the number of, we're going to reduce and destroy the vegetative cells of our microorganisms, but we're not going to be able to eliminate the endospores. It goes on to say exposing materials to boiling water for 30 minutes will kill most non-endospore-forming pathogens, including some resistant species, such as tubercle bacillus and staphylococcus. Okay, Probably the greatest disadvantage with this method is that items can be easily recontaminated when you remove them from the water. So then it's super important to dry things pretty quickly so that that water is not acting as essentially a vehicle, if you will, for inoculation by microorganisms again. Okay, so pasteurization. So some particular liquids, for example, do not do well when you boil them at 120 degrees Celsius. So think about milk. If you've ever tried to make a gravy, or maybe you made homemade hot chocolate and you had the flame a little too high on your stove, or the electric was too high, and so you ended up boiling your milk and it curdles, which is not a delicious feel in your mouth. So in this case with milk, some fruit juices, beer, wine, we are going to use pasteurization. So essentially, this is a technique, if you look at the last bullet, in which heat is applied to liquids to kill potential agents of infection and spoilage, while at the same time retaining the liquid's flavor and nutritional value. So we don't want to compromise the flavor of your fruit juice or the flavor or the taste of your milk, but we do need to radically reduce the amount of microorganisms on those substances. So we're usually thinking about a much cooler temperature, 63 degrees Celsius for 30 minutes. And this is for a big old batch of whatever it is, if it's beer or milk or apple cider. And essentially then it is going to dramatically reduce the number of vegetative cells, but it doesn't necessarily kill all vegetative cells. But what it does do is it eliminates the microorganisms that, look at the second bullet, that have the potential for spoiling the foods or causing illness. So we're not thinking about it being completely free of microorganisms, just being free of those things that are likely to cause the food to spoil or be harmful to us. So there are some other kinds of pasteurization you might have heard about. So we have high temperature, stable, wait, 1 second, HTST, high temperature, short time pasteurization. So the milk is heated at 72 degrees Celsius, so a little bit warmer, but just 15 seconds, and it's bottled and refrigerated. And then you can see there can be the ultra high temperature pasteurization. So this is milk that's 138 degrees Celsius for 2 or more seconds, and then sealed in these airtight containers that are shelf stable at room temperature. So there are different ways or different forms of pasteurization. And you can see here the milk-borne organisms that are killed by pasteurization, and these are the ones that we are the most concerned about. Okay, autoclaving, I know I mentioned this before, but essentially what we're doing is we're adding water to create steam, and the steam is going to increase the pressure inside the autoclave, so we can get up to 121 degrees Celsius, and we're usually doing that for about 20 minutes. So if we're making media here on the left-hand side, we see likely some sterile media. This one might be sterile, or this one might actually have some microorganisms in it. Nevertheless, we have to autoclave any media before we put it in our petri dishes or before we grow microorganisms. And then when we're done with them, so if this was a bacterial culture here, then we would need to inoculate them to kill anything that was growing on them, so we're not releasing that into the environment. Like I mentioned before, if you've had piercings, if you've had tattoos and so on, any metal equipment like that, or even like the dental, stuff they're using in your mouth, they are going to clean that and then sterilize that, so when you use it on the next person or patient, then they are free of microorganisms, so the equipment is sterile. So our moist heat methods, such as autoclaving, is essentially a way to completely remove all forms of microorganisms, including endospores, and so this is a form of sterilization. Okay, as you might imagine, incinerating medical waste, for example, the equipment like this is showing you the inoculating loop being flamed. So essentially, this is also going to kill all microorganisms even the endospores, and is going to reduce it to essentially ash. And we're talking about medical waste, is also going to reduce it to a gas.